Today on How It's Made, Archaea. It is true that most archaea don't look that different from bacteria under the microscope, and that because of the extreme conditions under which many of the species live, it has made them difficult to culture, so their unique place among living organisms long went unrecognized. It wasn't until the year 1977 that archaea were recognized as a separate domain from bacteria through the work of Carl Woese and his colleagues. They introduced the three main branches of evolutionary descent as archaea, eukaryote, and bacteria. There are three main types of archaea, and among these three types there are four subtypes of archaea. They are methanogens that produce methane gas as a waste product, halogens that live in salty environments, thermophiles, which live in extremely hot temperatures, and saprophiles that live in unusually cold environments.
Within the data table, it is shown that from the selected organisms they can be branched into three different categories or kingdoms based on the ribosomal DNA that was found in each organelle. The cell capsule comes before the cell wall, a sticky and slimy material that is made either of polysaccharides or proteins. This layer is known as the capsule. It generally helps to adhere to substrates or neighboring cells, and the capsule also prevents excess loss of water. Archaea contain a cell wall, a semi-rigid layer that helps the cell maintain its shape and chemical equilibrium. Although not truly unique, the cell walls of archaea are unusual. Whereas peptidyl glycan is a standard component of all bacterial cell walls, all archaea cell walls lack peptidyl glycan. The wall may be composed of only a surface layer proteins, known as S proteins. They form the S layer. Between the cytoplasm and the cell wall, there is a plasma membrane that surrounds the cytoplasm and its contents. It is a selectively permeable barrier, or mechanical barrier, of the cell. Nutrient and waste transport, location of many metabolic processes, and detection of environmental cues all happen here. Within the cell, there is a cytoplasm. This is where the living functions of archaea take place and where the DNA is located. Located in the cytoplasm, you will also find ribosomes, a nucleoid, and plasmids. The ribosomes are where protein synthesis takes place. The cytoplasmic ribosomes synthesize proteins destined to remain within the cell. The ribosomes of archaea and bacteria are the same size and are smaller than those of eukaryotes. However, more of the ribosome proteins, translation factors, and tRNAs of archaea more closely resemble their counterparts in eukaryotes. The next thing in archaea is the nucleoid, also known as the nuclear body, chromatin body, or nuclear region. It is an irregularly shaped region that contains the cell's chromosomes and numerous proteins. The chromosomes of most bacteria and all known archaea are a single circle of a double-stranded deoxyribonucleic acid, or DNA. For most bacteria and all known archaea, the nucleoid is a simple region in the cytoplasm. It is not separated from other components of the cytoplasm by a membrane. Plasmids are a genetic structure in a cell that can replicate independently of the chromosomes. A plasmid is a small DNA molecule within the cell that is physically separated from the chromosomal DNA and can replicate independently. Most commonly found as a small, circular, double-stranded DNA molecule within the cytoplasm. Pili are short, fine, hair-like appendages that are thinner than flagella. They allow cells to attach to surfaces and other cells, forming biofilms. Flagellum and archaea are used to move. They are thread-like locomotive appendages extending outward from the plasma membrane and the cell wall. Although the main function of the flagellum is mobility, they can have other roles. They are important for certain types of swarming behaviors and can be involved in attaching to surfaces. You now have a complete archaic cell. From here, the cells can branch out into extremophiles, which exist in extreme conditions such as hot springs or toxic environments.